feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome from very cold Singapore to the National University of Singapore Yong Loo Lin School of Medicine's Healthy Longevity webinar. We had a high today of 26 degrees Celsius, which is like the coldest I've ever seen. So I'm worried people are going to freeze to death tonight, but I think we'll make it through the show. I want you to remind you to use the questions and Q&A function to ask questions for the show. I'm sure there's some good thoughts in all of your heads, so let us know and we'll try to ask those. We have Professor Nazif Alic joining us tonight, and that will be a really exciting seminar. Before we get to that, we have Shija Navakode, who's a senior research fellow at our Center for Healthy Longevity, and she will talk about her own study. Alpha-ketoglutarate is a modulator of synaptic plasticity in Alzheimer's disease, a potential therapeutic approach.
Thanks for the introduction. Good evening to everyone. As mentioned, my name is Shija, and I will be talking about the role of alpha-ketoglutarate as a modulator of synaptic plasticity in Alzheimer's disease, which could be a potential therapeutic target. Alzheimer's disease is a progressive neurodegenerative disorder characterized by excessive accumulation of amyloid beta plaques, neurofibrillary tangles, hyperphosphorylated tau, along with synaptic and mitochondrial dysfunction, dysregulation of calcium homeostasis, neuroinflammation, etc. While the clinical symptoms of AD include progressive loss of memory that disrupts daily life along with many other behavioral complications. One of the earlier papers from Brian's lab showed that AKG promotes a longer, healthier life in mice by reducing the levels of systemic inflammatory cytokines. Chronic inflammation, particularly in the brain, has been linked to memory loss and cognitive decline, and any strategies targeting inflammation can reverse cognitive decline. So our hypothesis was if AKG can reduce the inflammatory cytokines, then it could possibly improve synaptic plasticity and memory and can extend the cognitive reserve of the brain. Therefore, the aim of the study was to see if AKG can rescue the impaired LTP and memory in Alzheimer's disease mouse model, or if AKG can extend the health span or cognitive reserve of the brain. In order to study this, we used a mouse model of AD called APPPS1, which contains a human transgene for both amyloid precursor protein and presenilin-containing mutations. We used a cellular model for learning and memory called long-term potentiation, or in short LTP, that can be induced in hippocampal slices. The x-axis represents the percentage change in field EPSP, while the y-axis represents the time in minutes. After recording a stable baseline, a strong tetanization was applied that leads to an increase in potentiation that persists for 3 to 8 hours. If it persists for longer, it's called late LTP, which is a correlate of long-term memory. Or if it decays, it's called early LTP, which is a correlate of short-term memory. LTP is found to be impaired in both males and females in APPPS1 mice. And you can see the decay of LTP is higher in the females compared to the male AD mice. We used a calcium salt of AKG called calcium AKG. When calcium AKG was bath applied for 1 hour during the time of induction of LTP, it rescued the impairment of LTP in both males and females. The bar graph on the right shows the comparison of male and female APP mice with calcium AKG. It shows a significant difference from 5 minutes until 50 minutes, indicating that the females shows a higher level of potentiation compared to males. In order to study the mechanism by which calcium AKG rescues synaptic plasticity in AD mice, first we checked the effect of mTOR inhibition by rapamycin. When rapamycin was applied to wild-type slices, it blocked late LTP, while application of calcium AKG along with rapamycin rescued the effects. This could be because mTOR inhibition might interfere with the synthesis of the required plasticity-related proteins. While in APP-PS1 mice, inhibition of mTOR via rapamycin rescued the impairment of LTP. Inhibiting mTOR would increase autophagy that could possibly increase plasticity and memory in AD mice models. Next, we studied the role of calcium-permeable AMPA receptor, which is a new perspective on the pathophysiology of Alzheimer's disease. CP-AMPA receptors are AMPA receptors without a GLU2 or those with an unedited GLU2. They could function to amplify the biological signals. Therefore, even a modest alteration in the expression levels of CP-AMPA receptors is expected to have a profound implication on synaptic transmission. When IEM, a CP-AMPA receptor inhibition, was bath applied during the time of induction of LTP in APP-PS1 mice, it had no effect, while along with calcium AKG, it failed to show the rescue effects, which shows that calcium AKG facilitated LTP involves the insertion of CP-AMPA receptors in APP-PS1 mice. To conclude, the AKG improves synaptic plasticity in APP-PS1 mice, and the rescue of LTP was more effective in females compared to male AD mice. Calcium AKG exerts its effect in AD by enhancing autophagy and CP-AMPA receptors, and for future work, this calls out for a gender-based approach for calcium AKG intervention studies in AD. And I would also like to thank Brian Kennedy and all the members of the BK Lab and the NUHS grant for the support. Finally, I would like to thank all of you for your kind attention.
Thank you, Shija. It's great to have Shija in the lab because having somebody that really has expertise in electrophysiology is very difficult to find. And so she's helping a number of projects move forward. It's my great pleasure to introduce our distinguished guest speaker, Professor Nazif Alic. His research delves into the fascinating world of gene expression and its impact on the plasticity of adult physiology and lifespan. He uses the fruit fly, Drosophila, as a model organism, and his work primarily focuses on transcriptional regulation, exploring both specific transcription factors and the basal transcription machinery. Today, he's going to share his insights on an intriguing aspect of his research, the lesser-known polymerases RNA-Pol1 and RNA-Pol3 in the aging process. His topic is aging and transcription in the fruit fly, the odd polymerases. Thanks for joining us, Nazif.
Thank you, and thanks for this opportunity to talk about our work. And a big hello from London, where we are scorching at 18 degrees. It's very, very hot here. So let me just share my slides. I hope you can see that. So as Brian said, my name is Nazif, and I work at the Institute of Healthy Aging at UCL. And my lab is interested in transcription and aging and how those two processes might interact, gene regulation and aging. So the reason why we're interested in aging is kind of the usual one, or at least the one that seems to be very important at the moment, and that is this increasing proportion of older people that we have in our societies due to the fact that we live longer. And this is kind of imposing pressures, both personal and societal, because age itself is the greatest risk factor for many diseases. And work by many very, very good researchers and sort of exceptional work done by Brian and others has shown in the last 30 years that aging itself is plastic. And this kind of gives this idea that you could target aging or processes related to aging to improve health in people. Now, that's kind of the overall ideas and the overall societal aim of what we do, but what we actually do is much, much simpler. We work in the fruit fly, and we are asking a very simple question, and that is what limits health and survival in old age. And by asking this question and getting the answers, we kind of hope that we will be able to describe what these processes that are related to aging are, or that are driving aging. But specifically in terms of experiments, what we do is we focus on identifying manipulations, for example, mutants, that outlive the wild type. So this is kind of different to other fields of biology where we're looking for a mutation that might impair a particular function. So a wild type that, a fly that can no longer have an eye. Rather, we're looking for flies that are performing better than what the wild type fly can do. And so this brings into question of what actually is a wild type. And we try to use an outbred healthy population of flies that are kind of very similar to what you would find flying somewhere in Europe, or specifically in Africa, actually, because our flies are from Benin. And we, in my lab, we are specifically interested in transcriptional gene regulation, so how you can regulate gene expression to change how an animal ages, to sort of slow aging processes down. And as Brian said, today I'll talk to you about the odd poles. And they're odd because simply they're called pole 1 and pole 3. So they've often been referred to as the odd poles. So the reason why we started looking at them, they're sort of often not studied in biology because they're thought to do this sort of boring function of just simply generating these RNAs like the ribosomal RNA or tRNAs that are not very entertaining for most people. So they've often been neglected. The reason why we started thinking about them is simply, it sort of starts with TORC1. And this idea that's been established in the field, that inhibition of this kinase complex is able to extend lifespan in a number of different species. So TORC1 is really well established as an evolutionarily conserved driver of aging. And this is in the field of the biology of aging. But when you look at the work that's been done on TORC1 in the field of cell biology, there's another process that TORC1 is going to regulate really tightly. And this is actually the biogenesis of translation machinery. So the biogenesis of the machinery that performs protein synthesis inside a cell. And so we started thinking about that, and especially because a lot of the work by Campbell and Kennedy labs and others has actually shown that some components of ribosomes, if you decrease their, can actually extend lifespan and delay aging. So for TORC1 to regulate the expression of protein synthesis machinery, it actually has to regulate all 3 RNA polymerases. So this includes pole 2 that will transcribe messenger RNAs that will give rise to ribosome, that will be translated into ribosomal proteins. But it also includes pole 1 and pole 3. And so this is why we started thinking that maybe pole 1 and pole 3 are also involved in aging, and maybe they're even acting downstream of TORC1. So I'll tell you a little bit about the 2 polymerases, or the 3 polymerases. And there's essentially these multi-subunit complexes where a lot of the subunits are unique to each polymerase. They're sort of paralogs. They perform the same function in the sense that they all transcribe DNA into RNA, but they will do that in different ways. They have different subunits, and they will transcribe different genes. So pole 1 really just has one task. It transcribes the ribosomal, the precursor to the ribosomal RNA. And pole 3, on the other hand, actually transcribes quite a few different RNAs, but most prominent examples are the 5S ribosomal RNA as well as tRNAs. So to start looking into these polymerases, what we did first is just obtain fly mutants that are mutated for a subunit that's specific to each polymerase. And so I'll just give you some examples of what we saw. And if you, obviously, if you completely lose the function of either pole 1 or pole 3, this is lethal. The fly can't develop and can't survive. But we could obtain heterozygous mutants, and specifically we're looking here at females, where the mutation results in a decrease of the transcript that's encoded by the gene of about 50%. And in each of those cases, both for POL1 and POL3, we could see that the females, adult females, are actually longer lived than their wild-type counterparts. So these are survival curves, and you can see that the POL1 heterozygous mutant as well as the POL3 heterozygous mutant are actually longer lived. Now, when you start looking at model organisms like C. elegans or Drosophila or even mice, you can actually see that it's sufficient to manipulate a particular process in only some cells in the organism, or only a specific organ, and this will then result in increasing the longevity of the whole fly in this case. So we tried to see which cell types POL1 and POL3 might be acting in, so which are the cell types in which their activity seems to be driving aging. And we very quickly established that the relevant organ itself is the gut. So you need to decrease POL1 or POL3 activity in the gut to be able to extend lifespan, or that's sufficient to extend lifespan. And what's special about Drosophila gut is that Drosophila itself is mostly post-mitotic, so the cells are not dividing throughout its lifetime. But it has one key organ, the gut, that is actually supported by stem cells. So there is mitosis that occurs and cells get replaced. And these cells are called intestinal stem cells. So by knocking down, again, a particular subunit of POL1 or POL3 in these stem cells, in adults, so starting from day 2 of adulthood, we could also show that this is sufficient to extend their lifespan. So it seems that actually POL1 and POL3 are driving aging, but specifically from the stem cell compartment in the fruit fly gut. Now, there are some differences, and one of the differences that we have observed is that POL1, if we knock it down in these mature cells, the enterocytes, the differentiated cells, we can also extend lifespan, whereas we've never found any evidence that reduction in POL3 activity in those stem cells can actually extend lifespan. So I'll focus from this point on a bit more on POL3, just because we've had more time to work on this. And what was the kind of interesting question that came out of this work is which specific transcript or group of transcripts might be required for the effect of POL3. Because as I said, it transcribes a number of different genes, including tRNAs, 5S, but then also some other short non-coding RNAs that are required, for example, for RNA modifications. And so to start looking at this, we first looked at biochemically, if we disrupt POL3, which RNAs are disrupted, which expression of which RNAs is actually affected. And so we did this in worms, or rather Jennifer Tullett from the University of Kent did this in worms. And what we saw is that the tRNAs were actually quite substantially affected, whereas the expression of the 5S and several other RNAs was not affected. And when we look at tRNAs, we actually see that it's not all tRNAs that seem to be downregulated in this long-lived POL1 loss of function worm, but it's actually specific tRNAs that are downregulated, and even more surprisingly, some of them seem to be upregulated, possibly due to compensation. And we see something similar if we look at the flies, where the strongest effects are observed on tRNA levels, whereas essentially no effects are seen on 5S and other RNAs transcribed by POL3. What is also quite surprising in the fly, and I can talk about this later if you'd like me to sort of explain what we think might be happening, is that we actually see a lot of the tRNAs, most of them are actually upregulated, with some of them being downregulated when looking at mature tRNAs. So it sort of seemed like the tRNA expression is what's important here. And so to query this genetically, to sort of try to find causality, we exploited the fact that different classes of genes transcribed by POL3 actually require different transcription factors to be expressed. So POL3 works off 3 promoters. It sort of uses one of 3 promoters to engage the DNA and start transcription. And those three promoters require different transcription factors. So one of the transcription factors that we didn't look at is TF3B, because this one's ubiquitously required. But TF3A is actually only required for the expression of the 5S ribosomal RNA. On the other hand, TF3C is required for the expression of both the 5S and the tRNAs. And a different transcription factor called SNAP-C is required for the expression of all these other RNAs that are produced by Pol3. And so what we did is we again obtained heterozygous loss-of-function mutants in specific subunits of these three transcription factors that allow us to differentiate between different genes transcribed by Pol3. And we performed a number of lifespans, so that we repeated them not at the same time, but sort of consecutively. So you can see that this actually took a few years to finish. But what we observed is that essentially loss of function in TF3C was able to robustly extend fly lifespan, whereas loss of function in TF3A seemed to sort of extend it sometimes, but sometimes not. The effect was not so robust. And so this is the survival curve of females that actually carry a heterozygous loss of function of TF3C. So this sort of indicated that you need to inhibit both 5S and tRNA to extend lifespan. And the fact that when you inhibit just the 5S, it seemed not to be robust or efficient, it did point to kind of reinforce this idea of what we saw before, is that it is actually the tRNAs that are relevant in this context. We also tried to see if we get broad improvements in health, or if these flies are just living longer but not feeling much better or not performing much better. And we compared here a loss of function in a Pol3 subunit to a loss of function in TF3C. What you will note that the name of this Pol3 subunit has changed. This is simply because fly nomenclature has changed in the meantime, but it is still the same subunit that we used initially. And so in flies, we can look at the gut barrier function as an indication of the health of the intestine. And this is essentially looking at how the gut becomes leaky as they get older. And we do that by feeding them a blue dye. So when the gut is leaky, they become blue, and this is what's called the Smurf phenotype. And so as you can see, the wild type, as they get older, they start their guts start getting more and more leaky. But both the Pol3 loss of function and TF3C loss of function, this phenotype is substantially decreased and delayed, indicating that both disruption of tRNA specifically and Pol3 are sufficient to improve gut health, at least this aspect of gut health. We can also look at the neuromuscular performance in flies, and we do that quite simply by inducing the negative geotaxis response, where they're trying to sort of run up a vertical surface. And how far they run in a given unit of time, as you can see for the wild type, it actually decreases as they get older. And for both the Pol3 and the TF3C mutants, this decrease is delayed, so they can preserve their neuromuscular function for longer. And so overall, this kind of indicated that disrupting tRNA expression seems to be sufficient to both extend lifespan and improve health in the fruit fly. What I'm not going to mention, but we did try to do a bit of work here, is that it does seem like this disruption of tRNA expression is causing a stress response, and it might be the stress response that's actually driving these health benefits. What I'll talk to next, though, is throughout this work, of course, and I started at the beginning saying that we're trying to use Drosophila as a model to understand what processes are driving aging, but the relevance, or something sort of that we want to refer to or refer back to all the time, is what might be happening in humans, because fundamentally it is human aging that we would really like to understand as much as possible. And so in this project, we did start thinking about what about human aging? Like, could this be relevant for human aging? And the reason why this was kind of quite confusing or not necessarily obvious is that we do know from human genetics or population genetics is that actually substantial loss of function in POL1 or POL3 subunit, also POL1, but POL3 subunits causes disease. And essentially you get these developmental disorders like ribosomopathies. Now, this is a substantial loss of function, and it sort of makes sense knowing, for example, that a full POL3 knockout in the fruit fly is also lethal. But we wanted to see if it's possible that a more moderate reduction in their activity might be related to aging, and specifically a more moderate reduction in the adult when the human's already developed and everything is fine. So we tried to do this in collaboration with a human geneticist called Carolina Kuchenbacker. And we tried to use human population genetic data to examine the relationship between natural genetically encoded variation in POL3 subunit expression. And we expect this to be quite minor because this is the variation you see within living kind of quite healthy, normal people. And to relate this to the genetic variation or genetically encoded variation in human longevity using a method that's called Mendelian randomization. So in the next few slides, I'll try to give you just a sort of better description of this method if I can, and just tell you what we think that we found, or what we found. And so we looked at this correlation between POL3 expression and longevity in specific tissues or specific organs, because expression is, there's expression in QTLs that we use actually organ specific. And we looked at organs that we thought would be important for longevity, such as brain, muscle, liver, small intestine, adipose tissue. And so what we did for these specific organs, we found expression in QTLs from GTEx, so this is all publicly available. And then for these same ones, we looked at what effect they have on longevity by using the summary statistics essentially from a GWAS study that was done by Joris Dillon and was published a few years ago. And in this GWAS study, they actually looked at the likelihood of people reaching or sort of living beyond the 90th percentile of their cohorts, so being in the top 10% longest lived individuals in their cohort, or being in the top 1% of the longest lived individuals in the cohort. So I'll explain a little bit more in a second about how what this relationship between expression and longevity looks like. But here, what I did want to point out is that we did find a significant correlation between the two. But this significant correlation was actually only present in one specific tissue, and this is the adipose. So it did seem like if the POL3 activity is related to human longevity, it's sort of coming from a specific tissue. We also did have a sort of strong signal from the small intestine, which would be the equivalent of the fly gut. But the number of eQTLs that we had here was too low for us to get anything significant out. So what do I mean by this relationship? But essentially what we see is that the higher the level of POL3 subunit expression, so this axis here shows you the relative level of POL3 subunit expression. The higher the level of expression, the lower the likelihood that an individual will live to be in the 10% of the longest lived cohort. So indicating that POL3 expression in the adipose is causally linked to a human longevity. And by causal, I mean this is causal in terms of how human geneticists talk. This is not necessarily causal how an experimental sort of geneticist would talk. We also found similar links for POL1 subunit expression and also for ribosomal proteins. But in each case, it's happened to be coming from a different tissue, and we don't understand why that might be. And we also, a simple explanation here could be that, for example, POL3 activity in the adipose might be driving a particular disease like obesity, but we couldn't see that. So we didn't get there was no strong evidence that these expression patterns are linked to just one specific disease. So I guess at the end, or sort of almost at the end, I'd just like to tell you what we think that we found. And I think the interesting thing here is that the biogenesis of translation machinery, and this includes the activity of Pol1 and Pol3 appears to be set for optimal growth and reproduction. So in sort of evolutionary terms, we all optimize to grow and reproduce as quickly as required. But these activities seem to come at a cost of longevity and health in old age. And we could find a little bit of evidence that this biogenesis of translation machinery might actually promote aging even in humans. A collaborator, Colin Selman, has actually looked at mice where he used a particular heterozygote mutant in Pol3. And what they found is that this particular mutation caused some benefits with respect to age-related conditions. So, for example, there was reduced osteoporosis in females, but overall, these mice were not longer lived. It could be that in mammals, Pol3 cannot kind of cause a loss of Pol3 might not be able to cause longevity, or it could be that we were simply looking at the wrong subunit mutant and something might be a bit off. So at the end, I just would like to thank all the people who actually did the work. So Sarah, who did all the human genetics, Danny, who started the project really in the flies, and then Guillermo, Yavuz, and Shahjahan, who did a lot of this later experimental work. And I would be very, very happy to take any questions, and I thank you for listening.
Thanks. That's an amazing amount of work there. And I guess one of the reasons I say that is because, we've done a lot of genetic studies in various organisms and, but we typically work with non-essential genes, having to make heterozygous animals and hoping you get a phenotype when you have hopefully 50% expression, but it could be different in different genes is a lot of extra work. And I guess related to that question, I'm wondering if that comes back to the tissue-specific effects you see in the human studies. If you're looking at a process like ribosome biogenesis or translation, there could be different rate-limiting components in different tissues. And so one reason you get effects in one tissue and not another is you're touching on that rate limitation in some cases and not others. Is that a reasonable hypothesis?
I think so, actually. And so when that paper was in review, before it went out, so as in when we submitted initially, we had this little thing where we showed a correlation between the likelihood of a tissue having a significant kind of effect and expression. So it seemed like the lower the expression of a particular component, the more likely it is to be relevant in that tissue, which kind of goes well with what you're saying is that each tissue might have a different part of the ribosome biogenesis machinery that's limiting. But the reviewers are very strongly opposed to that because most of the kind of p-values are not significant. So essentially, sort of they were saying we're just comparing zeros. So we had to take it out. But I think that's it's.
Well, now I wasn't a reviewer.
No, I think this is mostly human genetics sort of reviewers, very stats-oriented reviewers. But yeah, it does seem quite likely that a different component will be limiting in a different tissue. And also, I think that's possibly why we didn't see the effects with the mouse, because maybe that subunit is just simply not limiting in the tissue in which we need it to be. But, it might just simply also be that you can't extend mouse lifespan by modulating pulse rate.
Yeah, I wanted to go back to the flies for a minute, because you kind of almost alluded to this, but I don't think you directly said it. So, a lot of the yeast beans we found that were long-lived in the ribosome machinery were slow growing. So they, in the wild, they had a fitness defect. So the cells divide more times. They live longer in that sense, but they grow slower through the cell cycle, which would be a disadvantage in the wild. Were there fitness costs in these flies? I mean, are they smaller or some phenotype that make you worry?
Yeah, it is. I think that is quite interesting in the sense that these are heterozygous mutants, and as you saw, just one subunit is reduced by about 50%. And we do see changes in activity. So we can see that Pol1 or Pol3 activity is different when we look at the adults. But the adults that emerge through development, they're not delayed. They are like so minutely smaller that it's never significant. Like they are consistently smaller, but I think we've never found them to be significantly smaller. And their fecundity is perfectly fine. So somehow this kind of reduction is so small that it doesn't seem to cause any major problems. When we do an adult onset kind of intervention, when we do it in specific cell types, just in the adult, then actually you see even less of a difference. But it's really hard to tell though in the wild, because these are still animals kept under sort of, perfect conditions, living happy, healthy lives. So who knows what would happen in the wild? Yeah.
I was struck by the effect of the one mutant, the TF3A, I think it was, where you see a lifespan effect sometimes and not others. It doesn't all even look that great. I mean, it's a small n, but two times there was a big effect and two times nothing. What's causing the variability? Is it anything to do with the genotype, or is it something else that's going?
Yeah, so I think it's, we try to do the outcrossed sort of outbred population, just with the logic being that we don't want to rescue some sort of inbreeding defect. And flies are quite prone to that kind of inbreeding depression. And the lines of flies that have been kept inbred, they've been kept inbred for hundreds of years sometimes, well, hundreds of years. So they can be quite problematic. It does cause issues because it does become slightly crazy. We have to backcross them, but we're backcrossing into a outbred population, which then means that you can have selection, right? If your mutation starts interacting with something in the background, quite quickly you'll start getting selection. So it's not that we just backcross them. We often actually have to maintain them outcross, like every few months kind of go back and outcross them. So that can sometimes be a cause for variation, as you say, that on different times you might have slightly different background populations, or you might have left them, sitting there with not a massive population size for too long, and you just had selection, so that usually will annul the phenotype. But another possible reason for variation is that we use a complex mixture. So our food is actually yeast and sugar, and the yeast itself, we know from years of experience, it actually can have quite a strong effect as well, and that will vary simply from batch to batch. So it's never exactly the same. So my guess is that the reason why that TF3A is that there are certain conditions, either genetic or kind of nutritional, when it's enough, and then others when it's just simply not.
I always wanted to, and people have sort of tried this now, but not so much. I always wanted to do this experiment where you took yeast of different genotypes and fed them to flies and tried to determine the deletions in the yeast as food that would extend fly lifespan, and can get a better feel for the interaction there. I think it's a difficult experiment, but it's because the yeast deletions grow at different rates, and the matching them is not always easy, and there are problems. But it always seemed like a cool experiment.
I think it would be amazing to do. I think the biggest problem there is that we require a lot of yeast for food. Like the volumes are quite big. But some work like that has been done with C. elegans and E. coli. Yeah. And in fruit flies, Matt Piper has actually been doing a lot of work dissecting components of yeast, but essentially doing it by kind of generating this defined diet where you can actually take out different amino acids or lipids, etc., and see what effect they have.
If Fleischmann's is listening, this is a project they can sponsor. They produce yeast. Maybe that's a US thing. I guess I was a little confused because you had the worm data where most of the tRNAs, well, many of them were reduced expression and the worms were long-lived, and the flies where most of them are increased expression and the flies are long-lived. So what is it, up or down, or just dysregulated, or what's the story with lifespan?
So it seems like, my kind of final conclusion from that is that it's a dysregulation that's causing it. And I think sort of most of the evidence that we have points to that. When we saw that the flies are up, the fly expression levels were up, we were actually quite confused as well. what is actually going on. So what seems to most likely scenario, so the worms it's an RNAi that's applied from L4, and then we take them like 1 or 2 days later. So it's a reasonably acute intervention. The flies, it's actually the mutant, and we're looking at the mutant gut. So these things have developed with the reduced pulse reactivity for like 10 days, and then they've sat as an adult for another week. And so it looks like somehow they're able to stabilize mature tRNAs, because if we look on the other hand at precursors, so pre-tRNAs, the ones that we can see an effect, when there is an effect, it's a downregulation. So what's consistent between the flies and the worms is that it does look like pulse reactivity itself is down. It doesn't affect every tRNA in the same way. So some pre-tRNAs are down, some are not. But somehow the flies are able to compensate and stabilize their tRNAs probably, and you get this sort of skewing of ratios, but a lot of them are more abundant. On the other hand, though, what might not have been obvious from the graphs is that the scale at which this happened for flies is quite tiny. Like all the variation is around kind of a small, whereas in worms you have these massive downregulations and drops.
Yeah.
The upregulation in flies is actually quite small in magnitude.
Yeah. It reminds me a little bit of a study we did a long time ago with rapamycin in mice, where we found that if you treat the animals with rapamycin, you get the day you treat them or the day after, you get this big effect on translation. So we're looking at just translation generally by polysomes. But if you keep doing that every other day, and the day after the month when you do it, there's no change in translation relative to controls. So there's compensation going on, and I guess our thought process there was that it must be changes in, cap dependent or independent translation or other specific changes in certain messages being translated that was driving the long-term phenotype, but we never really went past that.
So my kind of feeling in our case is what might be driving the long-term phenotype is this disruption of ratios in tRNAs that might be causing kind of like a mild stress for the cell.
So it's invoking the stress response pathway downstream. That could have been happening in our context as well. And we certainly saw evidence of things like GCN4 pathway, which is, well, I forget what that is, XBP1. No, that's not it. It's
ATF4.
ATF4, yeah, that's right. I'm losing my mind. So we saw that pathway being invoked. And so, it could have been similar stress response pathways in our context as well. I'm curious about the tissue specificity. It's like, why adipose? I mean, is it just hitting the right expression levels, or, because this is not an extremely proliferative tissue. In the fly case, if you're hitting the proliferative tissue, and for translation, that could make sense, but adipose is not a highly proliferative tissue. So is it just the sweet spot, or could there be something specific going on there?
I mean, my honest answer would have to be I've got no idea. But I think one thing to realize with GTEx is that what it's identifying is probably enhancer elements. And it's possible that, the entire lineage might share certain enhancer elements. So when it sort of goes adipose, you might be thinking adipocytes, but it might actually be, for example, pre-adipocytes or some other. So I think I kind of try to think of it like that. It's sort of probably defining enhancers. Now, why we're only seeing it in this tissue, I'm not sure. All the tissues that come out, when you look at POL1, POL3, and even ribosomal protein expression, they're metabolic. It's sort of the tissues that you would think in a mammal are kind of very important for metabolism. Maybe it's something to do with that. Maybe they're able to have systemic effects by somehow moderating either some signals or overall metabolism.
So what's next? Where's the project going?
I think we still, like with POL3, we have sort of more curious findings at the moment where we're not completely sure that low activity is always better. And I guess the idea is now to try to see if there is sort of cell type specific differences in what's required for aging with respect to Pol3 activity. So that's kind of where this project is going at the moment.
And also the Pol1, you're still following up on Pol1 as well.
Yeah, for very silly reasons that is always delayed. It's always like about 3 years behind Pol3. So we are now kind of at the stage of trying to work out what exactly might be causing the longevity. Is there another kind of stress response, something like that happening? Is there are there changes to translation? What exactly is going on? So give us another few years and we might be able to tell you.
I'm going to hold you to that. So, have you looked at nucleolar size in these mutants and cells? Because there's a lot of thought about ribosomal processing and nucleolar size and aging as well.
We have in the Pol1 mutants, and we do see, so for example, because we focused on the gut, because genetically that seems to be where it's all happening. And we do see changes in nucleolar size. Weirdly, though, we only see them, so we see a reduction in nucleolar size, but only in the cells with the big nuclei, which are the enterocytes, so they're the differentiated cells. We didn't see a reduction in the stem cells.
There's a lot, I assume there's more translation going on in the enterocytes.
Yes, yeah.
Yeah.
And there's probably much more ribosomal RNA expression. They're highly polyploid, so they'll go like masses of everything. The nucleus is quite big. So yeah, it seems that inhibiting Pol1 will reduce nucleolar size, but in this case, specifically in enterocytes, not so much in the stem cells.
Great. And I want to bring in Curie, who's a research associate in our Center for Healthy Longevity, to bring you any questions from the audience. And by the way, ask questions. I'm sure some people have better questions than I do out there. So let us know and we'll convey them.
Thank you so much, Prof. Brian. Hi, Prof. Nazif.
Hi.
We have questions from our audience, but maybe you have already stated once, but it's about, does the Pol3 reduction slows down the stem cell divisions?
So we can look at this. What we tend to do in the fly is just a steady state of number of cells that are dividing at that particular point in time. I mean, we can look at young flies and older flies. And what happens in older flies, you had this hyperproliferation and misdifferentiation. So you get this hyperproliferative phenotype in older flies. The Pol3 loss of function, when they're young, they seem pretty much the same as the wild type. They have some division and there's no difference there. When they get older, the wild type sort of gets a lot of divisions and it's looking not healthy. The Pol3 limits that increase. Pol3 loss of function limits that increase. So basically, it looks like in young flies, not much of an effect. In older flies, with respect to this hyper-proliferative phenotype that you get with age, it does reduce the severity of that phenotype.
I see. Thank you. I have a few questions for myself. So in terms of like, how could partial inhibition of the fundamental transcription process be balanced to extend the lifespan while avoiding the adverse effect on maybe essential cellular functions?
I think what we are seeing is that this reduction is actually quite small, and that seems to have a positive effect. But at the same time, it's also, I guess, cell type specific. So, in a particular cell type, it might not be required as much to maintain. So, for example, like when we do it in the stem cells, they don't lose their stem cells. They're sort of still there. They still seem to be able to function. There was something else that popped. Yeah. No, but yeah, also, I guess the idea being is that simply, there might be the level of activity might be dictated by what's required for development to reach reproductive age. And then after that, these cells might not actually need as high an activity as they do before.
You mentioned stem cells, and I thought I'd just kind of add to that question a bit. what about like organoid models or in vitro stem cell models? Is there value to studying these models in terms, they're difficult to assess in terms of aging, but it could be an interesting way to get into human models and see what the effects of these mutants are.
Yeah, it is super interesting. We haven't done any, and I'm not sure if we will, because it's sort of slightly outside of, maybe one day we might start thinking about that. Other people have. So there is, for example, some work done on Pol3 in adipocytes, which I could have actually mentioned when we were talking about adipocytes, where you actually need to modulate Pol3 activity for differentiation into an adipocyte. So it's not simply that the pre-adipocyte needs Pol3 to survive, you actually have to moderate the activity to differentiate. So the involvement of these kind of what's usually is sort of considered as a like a stable just housekeeping process, quite often actually seems to be a bit more subtly kind of built into different decisions that a cell has to make, or yeah. Sorry.
I've reached the opinion that there's no such thing as housekeeping. You give evolution 3 billion years and it'll find all kinds of regulatory things to do with so-called housekeeping genes. It's fascinating how complex the system gets.
No, yeah, I totally agree. I think sometimes also the opinions that we hold as biologists come from this bias of thinking something is housekeeping and hence, can't do something else.
Yeah. Aaron, are there more questions?
Maybe I can go with this one. Is there any interdisciplinary methods or innovative technologies that we can bridge the gap between fruit flies model and human aging research, especially when we are targeting this kind of basic cellular process like transcriptions?
I'm really pro human genetics in the sense of doing population genetics, because there's so much data out there. So you don't necessarily need to get more data sometimes. You just need to think of a better question almost, and possibly, get better statistical or computational methodology. And like, I'm not a human geneticist. Whenever we do this, we have to do it with a collaborator who tells us what we can and cannot do. But I think that's sort of almost like an untapped potential. And just thinking about the data in different ways, processing them in different ways, but then also being slightly kind of more attuned to what is relevant and what is not relevant, because sometimes there's a risk when you have a lot of data that you just, you use it for things that you can't be used. Like, you start comparing apples and pears, and I think there's a risk there. But I think for me, that seems quite exciting.
Yep, exactly. We need more explorations for that thing. So we have one. In what ways does the disturbance of the tRNA levels specifically contribute to the longevity phenotypes observed in fruit flies?
Yeah, so we're not completely sure, but we sort of see two things. One is if we look at the whole fly, we do see that there's some sort of looks like differential translation. So when we do riboseq on a whole fly, we can get things that look like they're differentially translated in the mutant versus the wild type. So there could be what Brian mentioned, this idea that certain transcripts are getting better or less well translated. Another thing that we see is that there seems to be an activation of a stress response. So it's possible when the cell doesn't have the correct levels of tRNAs, the ribosome might not be happy or might be stalling or crashing into each other or something like that. And this seems to trigger essentially a proteostatic kind of response. So we have things like XBP1 upregulation and things like that, which seem to point that it is this stress response that might be driving the longevity phenotype. However, I'm unable to tell you conclusively because we simply just never did epistasis experiments to really.
That was my question.
Yeah, Janie tried to do them in worms, and they were just such a nightmare. Nothing like she looked at ATF4, ATF6, and XBP1, and none of them on its own was required for pulse longevity. Is it because of redundancy? I don't know. So my feeling is that it's probably there is some sort of a stress response and it's contributing, but we've never been able to explicitly show, like through nice, neat genetic epistasis, that this is what's happening.
So how do this finding integrate with existing theory of aging, like such as the roles of cellular senescence and metabolic regulations?
Yeah, I guess sort of the theories that you mention is role of cellular senescence and metabolic regulation. So I guess you mean the mechanistics, like how does it fit with the mechanisms that we think might be promoting aging or causing aging?
Yeah.
And I guess in that sense, I sort of think of it as sitting at the level of proteostasis. And this sort of balance between production and degradation of proteins and sort of how you maintain their function. I guess that's kind of where I think it's probably sitting. But on the other hand, that's a cellular process which might have, consequences for the tissue, which might have then, endocrine signals going off in all different directions, changing the physiology of the whole organism. And that's possibly what's happening in the fly gut, is that if you, change the behavior of stem cells, that might change the physiology of the whole organ, and the enteroendocrine cells, for example, might start secreting different factors. So I think simplifying it to just saying this is proteostasis, it's probably not right.
Probably worth making the point that people may not realize is that making ribosomes is an exceptionally expensive thing for the cell. And these are some of the most highly transcribed genes in the cell, and the cost of making ribosomes is an ATP is extremely expensive. So where you might have a lesser process and you make very subtle changes, it may not have much of an effect. A subtle change here in ribosome efficiency or levels or subunit composition could have a big effect on cell because of the amount of effort that has to be put into keeping it regulated in equilibrium. Maybe I can go ahead. I'll let you comment on that. Then I'll ask you.
Yeah, I was just going to say there was a very nice paper on POL1 where they showed that inhibiting POL1 can also extend lifespan in worms. And they go into much more detail thinking about sort of energy balance and how slightly reducing POL1 actually can liberate a lot of energy. And they go substantially further, I guess, than we did in our paper into potential mechanisms.
Interesting. Last question. How do you translate this?
I don't know. I'm hoping that somebody else
drug or some sort of gene therapy. What's the
I don't know. I mean, the polymerases themselves are druggable, and people are thinking about drugging them, for example, in the context of cancer. So eventually there might be, a potential to drug them. I think for the moment, we're just very focused on trying to understand what the basic kind of principles behind these processes that drive aging are. I think once we understand them, we'll also understand at which point to intervene and how.
Well, thanks, Ian, and thanks, Nazif. It's great to have you on. I'm waiting for the next POL1 result. We'll have you back. But thanks for coming on the show, and I hope you survive balmy London.
Thank you. It's been really great. It was a pleasure to be here.
I want to remind participants to use the chat function and panelists and all attendees option to leave comments and feedback on the show so everyone can see what we're thinking. We have another healthy longevity webinar April 3rd by Austin Argentier, who's a research fellow at Harvard and the Broad Institute. And right now he's keeping his topic a mystery. Finally, on the final video, I want to leave you with Anne Hathaway, who shares some of the most inspiring advice on aging. It's a beautiful reminder to embrace every stage of life with joy and confidence. You won't want to miss the uplifting message or the next show on April 3rd. And thanks for joining us tonight.
Academy Award
award-winning actress Anne Hathaway has been a force in Hollywood for more than 2 decades. The movie star is known for her wide-ranging talents both on and off the big screen, from being an advocate for women's rights to a fashion icon. And at 40 years old, the mother of 2 is embracing who she is. So a couple of years ago, you said,
What did I say?
when I look back on my 20s, I just remember being afraid of everything. And in my 30s, I'm actually excited about things.
Yeah.
So now you've entered your 40s. What would you say about it so far?
I'm cherishing. I'm right at that point where I have a much better sense of how I like to do things. I'm so much better at sharing. I feel like I'm kinder to myself. I feel like I'm kinder to others.
Growing up as a teenager in Hollywood, Hathaway quickly rose to fame, becoming a household name.
I started in this business when I was 17 years old, in a very different era. And there was this perception that there was going to be a cliff. And that cliff was a really young age. And the world's changed since then.
We're all starting to say, wait a minute.
Hang on.
Hang on.
Hang on. What about what I think? What about what I think about what I want to do? I don't know how you feel, but do you feel like you're getting better?
Are you kidding me?
Like,
we're just getting started.
You know?
No,
Thank
absolutely.
you.
And I wouldn't trade anything. For Hathaway, age is nothing but a number. Let's say on social media, I post a picture, or you post a picture, and people say, oh, you're aging gracefully, or oh, you look so good for your age. Do I say, thank you? Are we leaning into the hype of aging?
I don't think about age. I don't.
Tell me.
To me, aging is another word for living. So if people want to pay you a compliment, it's nice, but also whatever the hype is, I'm interested in what's beyond the concept of hype.
Today, more than 20 years into her career, Anne Hathaway doesn't take anything for granted.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.